The next high energy molecule is the thioester containing molecule. Oftentimes, for the sake of this course, we're talking about molecules containing coenzyme A or CoA. Pictured on this slide is a picture of acetyl-CoA. CoA is oftentimes used to move around acyl groups or acetyl groups, especially you'll see this during lipid synthesis, for example. Let's talk about the molecule to kind of orient ourselves here, folks. Most of what you're seeing here on the screen is CoA, and very little is the acetyl. So acetyl-CoA has this nitrogenous base and sugar, some phosphates, and a large organic group as well. All of this kind of here is the CoA part. Right here, this sulfur-carbon bond, this is the thioester, high energy bond. And then you can see, hanging off on the end, a tiny part of this molecule is the group it carries. In this case, it's an acetyl group. The hydrolysis of the thioester bond is slightly more exergonic even than hydrolyzing an ATP. And this is because the orbital overlap between the sulfur and carbon atom is not ideal. We will see how cleavage of this bond releases energy. For example, when we get to the citric acid cycle or TCA cycle, we'll see that hydrolysis of the thioester bond results in the synthesis of a high energy compound, GTP. Let's try to understand through drawing in a little bit more detail what's going on to make the CoA so high energy. And I want to compare the thioester bond to a regular old ester bond. So here is an ester bond that you all should be familiar with, right? What's happening with this bond is that, excuse my cartoon drawings of orbitals, we have this kind of ideal orbital overlap between the oxygen and the carbon. This allows for resonance stability of this functional group, right? We know that more resonance means more stability. So the ester bond is very stable. Let's compare that to the thioester bond. The orbital overlap between the sulfur and the oxygen is not ideal. Sulfur is slightly bigger, right? And because the overlap is not ideal, sulfur is actually less able to resonate into the carbonyl. And because it can do less resonance, it is less likely to do the same thing. It is therefore less stable. Things that are less stable are higher energy, so cleavage of this bond here should release more energy. Last up, folks, we will talk about redox molecules as high energy.